Acute gastroenteritis is a common cause of morbidity and mortality worldwide. It consists of inflammation of the mucosa of the stomach and the intestine. Its symptoms may include diarrhea in addition to nausea or vomiting. Noroviruses are a leading cause of acute gastroenteritis. Every year, 120 million people around the world are infected by norovirus, and approximately 35,000 of them die. Noroviruses belong to the Caliciviridae family. The genome of the members of this family is a positive single-strand RNA molecule. For this reason, they belong to Group 4 in Baltimore's classification of viruses. Recently, a new genre has been described in this family, Recovirus genera. The RNA molecule is organized in 3 open reading frames. ORF1 codifies for a polyprotein that is processed in 7 non-structural proteins. ORF2 codifies for the principal VP1 protein, and ORF3 codifies for a structural VP2 protein, highly divergent. Norovirus are non-enveloped small round viruses surrounded by an icosahedric capsid. Their diameter is about 27 to 40 nanometers.
The capsid is composed of 180 copies of the VP1 protein.
This protein can be divided in P2 subdomain, which extends above the surface, P1 subdomain, and S domain. Also, it presents an N-terminal extreme that forms the inside of VP1 protein. The 180 copies of the protein are structured in 90 dimers. Noroviruses are classified in 6 genogroups. Human strains are classified in genogroups 1 and 2, and 1 genotype of the genogroups 4 and 6. Animal strains are classified in the rest of genogroups. Genogroups 1 and 2 are subdivided in 9 and 22 genotypes respectively. Genotype 4 of genogroup 2 is the most predominant strain. The 2 main obstacles in the study of human norovirus are the lack of an in vitro culture system that supports the replication and of an efficient animal model which mimics the disease. Baylor College researchers provided a solution to this problem. They replicated human norovirus in a 3-dimensional in vitro culture system based on human intestinal enteroids. The enteroids imitate the intestine. They include its characteristic domains, crypt domain and villus domain, as well as all cellular types of the intestinal epithelium. At the bottom of the crypt domain we find the intestinal stem cells, from which enteroids are isolated. These cells are intercalated with Paneth cells, which establish the stem cells niche. Above these cells we find the transit amplifying cells, which give place to the mature cells, including enterocytes, goblet cells, and enteroendocrine cells. This recent advance will contribute in the further study of the norovirus's biology. The development of efficient methods for norovirus infection prevention and treatment may be possible in the following years. These will reduce the occurrence of gastroenteritis caused by those viruses.